Hello, and today is our first lecture in cycle of tropical disease. There will be 3 parts of it, and we'll start with features of pathology of the tropics, classification of tropical disease, peculiarities of course of infectious disease in countries with tropical climate. Tropical medicine, part of medical science, studying the questioning of theory and practice of health protection in tropical countries. There are some specific problems. Development of studies about tropical illness, questions of hygiene of tropics, organization of health care in the tropics. What is the tropics at all? Yes, tropics is a part of the Earth's surface located in the equatorial, subequatorial, tropical and subtropical climatic zone between 23 and 5 degrees of the northern latitude and 23 and 5 degrees of south latitude. This zone includes Africa, Australia, India, Indochina, Indonesia, Iran, Iraq, Pacific Islands, South and Central America. Also, equatorial area where is constantly high temperature, 24-28 degrees, abundant rainfalls, in some places up to 10,000 millimeters, thick multi-leveled forest, variety of flora and fauna, which may be the vectors or the source of infection of different diseases. Subequatorial zone. Constant high air temperature 22-25, change of dry and wet seasons, dominant of savanna, deciduous and evergreen forests, deserts and semi-desert, like in Sudan, Chad and Mali. Tropical area. Temperature in winter not below 10, in summer 30-35. Distinguish the season of rains, summer, and season of drought, winter. Desert, semi-desert, savanna, deciduous forest. It's Libya, Algeria, Egypt. And subtropical area. Alteration of moderate in winter and tropical in summer. Temperature condition. Monsoon express differences in the rainfall. Desert, semi-desert and evergreen forest. Features of tropical climate. High sums of annual heat. Oscillation of fallouts in the tropical forest within 3,000–5,000 millimeters per year. In desert up to 200 millimeters per year. Fluctuation of temperature about 40 degrees in a day. The large temperature group and freezing in mountains. The features of tropical pathology depends on the environment conditions and social factors. So both play a role. And environment condition is something we can't control and something that will actually changes in the latest year, and social factors are kind of controlled, but it's a big work. It's not something that you can change in a day. Factors of the natural environment. Temperature, direct influence, increased water consumption and high risk of intestinal infection, violation of mechanism regulation body temperature and electrolyte balance, disorder of water salt metabolism, lower acidity of gastric juice and decreased barrier function of the stomach, high frequency of colds, temperature variation and decreased barrier function of the upper respiratory tract. Indirect influence. Wide distribution of numerous kinds of insects, including blood-sucking, that can be carriers or so-called vectors, of infection. So somebody who transmit disease from one to another. So we see Anopheles, phlebotomous mosquito or sandfly and others. A hot climate assists the height of different types of plants and animals. So it's favorable conditions for existence of heat-loving causative agent of infectious and invasive disease, viruses of yellow, dengue fever, filariasis, optimal habitat for development of gout, gelmins, ascariasis, trichoris, and others, high variety of special vectors dwelling in a temperature climate, Anopheles mosquitoes. Existence of some vectors is possible only in the conditions of tropic, like tsetse flies, kissing bug, and others. So the disease are spread only in tropical countries. Due to this, it's connected with vectors. Again, another big problem is water. Reproduction of causative agent and preservation of them in the water, cholera, typhoid, dysentery, amoebiasis, leptospirosis, shellfishes, schistosomiasis, crawfish, cyclops, radiculiasis, fishes, opistrochiasis. Development of special vectors, mosquitoes, malaria, filariasis, yellow and dengue fever, midgets, and others. The role of water increases on the next reason. Swimming in open reservoirs, they may be risk of schistosomiasis, for example, just swimming. Using of the reservoirs with a technical purpose, drinking of unboiled water by majority of the population, use in food of raw water plants like mint, chestnuts, nut, lotus, fish, crustaceans, shellfishes. Contamination of the river, ponds, wells, soils by microbes and eggs of helminths in the rain period. Increasing concentration of the contagious material in reservoirs in the period of drought. Soil. Another factor is source of developing helminths, primary cause of disease, agricultural workers. A reservoir for multiplication of insects transmitting infectious disease illness, solar radiation, high frequency of sunburn and skin cancer, irradiation assists the sanation of environment. Socio-economical factors. Low level of sanitary culture and illiteracy of population, taking of unboiled water is a reason of half of infectious disease, absence of the sewage system, dirty hands, bad housing terms. High death rate, especially among children. High birth rate, like 4-5% in the year. Insufficient nourishment in albino starvation, hypovitaminosis, and pellagra. Occupation of the population also play roles. Leptospirosis and helminthiasis more often registered among the peasants. Brucellosis, anthrax, echinococcal disease among nomads. Lodgers and hunters as a rule suffer from yellow fever and cutaneous leishmaniasis. Plantation workers from larvae. Sorry, technical problems, so I'll maybe repeat a bit. So, lifestyle, dietary habits, superstition and customs, like presence of blood in the boy's urine in schistosomiasis is regarded as evidence of sexual maturity. Underdeveloped medicine, centralization of hospitals in cities and their absence in the villages, the remoteness of villages from each other, impairs the provision of medical assistance. 10,000, 60,000 persons per one physician in rural area where that is rather low level. Medical features of tropical disease. Millions of sick people. Filariasis, 250 million, schistosomiasis 270 million, leprosy 12 millions. Infectious tropical disease, non-infectious tropical disease, and tropical disease of unknown origin. These are the groups that are available, like infectious tropical disease that are registered only in tropics and outside the tropics. Non-infectious tropical disease connecting with the direct action of the environment, connected with the effects of natural and social factors, genetic disease, other therapeutic disease. Tropical disease of unknown origin, like cardiac fibrosis, cardiomyopathy, Kaposi sarcoma. Well, now it tends to be like virus caused, malignant tumor of lymphatic system. Infectious tropical disease, registered only in tropics, like viral yellow fever, dengue, Lassa, Marburg, Ebola. Spirituosis, Yaus, Pinta, tick-borne recurrent typhoid, bacterial Buruli ulcer, registered in and outside the tropics. Viral, measles, chickenpox, influenza, acute respiratory viral infection, rabies, polio. Spirituosis, leptospirosis, loose-borne recurrent typhoid, bacterial, cholera, plague, salmonellosis, dysentery. Helminthic, ascariasis, enterobiasis, tiniosis, Gymnolipidiasis, Diphyllobatriasis, echinococcal disease, Pistochiasis, that is in and outside the tropics, fungus, Trichophytos, protozoal, vivax malaria, Balantidiasis. That is registered only tropic. Guinea worm disease, Filariasis, Schistosomiasis, fungus, Mucormycosis, protozoal, tropical malaria, Leishmaniasis, Trypanosomiasis. Peculiarity of the course of the tropical disease. Unfavorable background, the deficit of power, hypovitaminosis and others. Massiveness of the infection. Early age of primary infection. The direct influence of the original conditions of the environment. Possibility to get several species of parasites. Polyparasitism, 2, 3 and more worms or worm infection with bacterial infection. So it will mean in the treatment later. Subclinical or obliterated course of many diseases in the population of endemic foci due to constant contact with pathogenic microorganisms and the development of immunity. Severe course of the disease like meningococcal infection, measles, tuberculosis and others. Chronization of many disease. Unusual course of self-infection like skin diphtheria often and pharyngeal diphtheria rarely, which is vice versa in European countries. Presence of some diseases prevents contamination of other diseases, like anemia S and C prevents malaria infection. Combination of some infections provokes development of tumors, like Epstein-Barr virus plus malaria provokes Burkitt lymphoma. And so this leads us to features of work of the doctor in the tropics. Like first, in all cases, unclear fever, appoint anti-malarial drugs. In the treatment of all infectious disease, simultaneously appoint anti-malarial drugs. Before a surgical operation, appoint anti-malarial drugs. Before the surgical operation, examine the patient at helminthiasis and treat him. After revealing a single parasite, continue to search of other pathogens. The most important disease of the tropic according to World Health Organization: malaria, filariasis, leishmaniasis, schistosomiasis, trypanosomiasis, leprosy. Measures of health protection in the tropics. Before traveling to tropical countries, is necessary total medical examination. Vaccination against typhoid, paratyphoid, tetanus, polio, yellow fever, cholera, hepatitis A. Follow measures of food hygiene, boiling, chlorization and filtration of water, general processing of vegetables and fruits. Chemical prophylaxis of malaria and sleeping sickness in West and Central Africa. Protection from the bites of insect, mechanical protection, canopies, impregnated mosquito repellents, measures of chemical protection, the use of insecticide and repellents. Well, that's the end of the first part of the lecture, and we'll continue with the second part. Second part of the lecture, trypanosomiasis. Introduction. The name is delivered from Greek word trypano means borer, soma means body. There are unicellular flagella protozoa, have spiral-like motion, need more than one host to complete its life cycle, often transmitted by a vector, generally found in intestine, but sometimes found in blood stream or in heart. Trypanosomiasis. West African trypanosomiasis and East African trypanosomiasis, caused by Trypanosoma brucei and rodensi, cause sleeping symptoms transmitted by Glossina, tsetse fly. American trypanosomiasis, caused by Trypanosoma cruzi, cause Chagas disease transmitted by Triatoma or winged bug or kissing bug. Morphology of trypanosome exists in the two interchangeable forms: trypomastigotes in blood, lymph, tissue, space of various organs and CNS is terminal and fatal. Epimastigotes in salivary glands, olfactor and culture media. Trypomastigote: single-shaped, central nucleus, free flagellum, undulating membrane. Long slender form, short stumpy form, and intermediate form are present. African trypanosomiasis or sleeping sickness belong to a group of closely related trypanosomes in the Trypanosoma brucei species complex. Three morphologically indistinguishable species are recognized. Some authors consider these as subspecies. Trypanosoma brucei, infect game animals and livestock, cause Nagana. Trypanosoma brucei rodensis, cause East African trypanosomiasis. Trypanosoma brucei gambians, caused West and Central African sleeping sickness. Distribution, like East African trypanosomiasis and West African trypanosomiasis. Glossina papalis and Glossina mortiscens are vectors. Trypanosoma brucei species. West Africa. Trypanosoma brucei gambians, less plentiful, cannot live in lab animals, reservoir host goats, cattle and pigs, transmitted by Glossina palpalis. East Africa. Trypanosoma brucei rodentia, more plentiful, can live in lab animals, nucleus is shifted posteriorly, reservoir host wild game animals and antelopes, transmitted by Glossina morticens. Major differences. The vector mortiscens group in Rodentia, palpalis in Gambians. Ecology, dry brush woodland in Rodentia, rainforest, riverland, lakes in Gambians. Transmission cycle, ungulate fly man in Rodentia, human fly human in Gambians. Non-human reservoir, wild animals in Rodentia, domestic animals in Gambians. Epidemiology, sporadic safaris in Rodentia, some epidemics in Gambians. Disease progression, rapid, often fatal in Rodentia, slow, approximately about a year, acute, goes to chronic form in Gambians. Parasitemia, high in Rodentia, low in Gambians. Asymptomatic carriers, rare in Rodentia and common in Gambians. Transmission, a bite from infected tsetse fly causes African trypanosomiasis. Blood transfusion, a rare cause of parasitic transmission. In rare cases, accidental transmission in the laboratory lab has been implicated. Life cycle. So it goes into fly, then has its cycle there. Again, fly bites a person. Then in human, metacyclic trypomastogote transform into blastoid trypomastogote, which are carried to oocytes. Trypomastogote multiply by binary fission in various body fluids like blood, lymph, and spinal fluids, and then trypomastogote again go into the blood and cycle repeats. Pathogenesis. Incubation period about 2 weeks in common. Trypanosoma chancre at the site of a bite. Spreading via lymphatics, enlarged lymph nodes, especially posterior cervical region with the bottom side. Then spreading via bloodstream. Headache, fever, fluctuating muscle and joint pain, irregular erythematous rash, invasion of bone marrow, hypoplastic anemia, enlarged liver and spleen, generalized weakness. Invasion of CNS, severe headache, mental apathy, slow speech, deep sleep, coma, and death. In East African trypanosomiasis, disease runs more rapid and fatal course. Clinical presentation. Incubation period may be from few days to weeks. The first clinical manifestation of African trypanosomiasis chancre occur at the site of inoculation. So these are trypanosomiatic chancre. Progression of African trypanosomiasis. So in Gambians, this takes months to years. Is Rodentia, this first may be absent, first clinical sign, and in weeks to months, death is coming. So first, chancre is appear or don't appear, then intermittent fever and headache appear, then through the lymphatics it spread continuous febrile episode, lymphadenopathy, deteriorating health, and then damage CNS. And first appear anorexia, minor neurological symptom, apathy, lassitude, then severe sleep disturbances and severe neurological symptoms comes, and adds with convulsions, coma, concrete infections, and death eventually. Clinical presentation. Diagnosis. Clinical picture plus demonstration of trypanosomes, like confirmation. Microscopic examination of unstained or stained blood films, culture on suitable medium like NNN, animal inoculation. Diagnosis. General anemia, hyperglobulinemia, low complement levels, elevated ESR, thrombocytopenia, hyperbilirubinemia, but not eosinophilia or abnormal liver function. The total IgM level is high in blood and CSF. A definitive diagnosis of infections requires actual detection of trypanosomes in blood, lymph nodes, CSF, skin, chancre aspirates, or bone marrow. However, symptomatic improvement after empiric treatment is the usual confirmatory test in areas where diagnostic stage studies are not readily available. Imaging studies. CT scanning and MRI of the head. Both head CT scanning and MRI revealed cerebral edema and white matter enhancement, respectively, in patients with late-stage African trypanosomiasis. EEG in neurological involvement usually shows slow wave oscillation, delta waves, and non-specific finding. Treatment. For early stage, we use pentamidine for Gambian's disease and Suramine for both Gambian's and Rodensky disease. For late stage, we use eflornithine for Gambian's disease and melarsoprol in Gambian and Rodensky disease involving CNF. It has its own drawbacks, each of them. American trypanosomiasis. So it's spread in South America. Trypanomastigote slender-shaped, central nucleus, C or U-shaped, free flagellum 1/3 of body, large bulging peripheral kinetoplast. A mastigote, obligatory intracellular mainly in cardiac and skeletal muscles, brain meninges, nerve ganglia, cells of GIT, etc. Epimastigote is present in vector-only, single-shaped kinetoplast anterior to central nucleus, undulating membrane, its short terminal free flagellum. So its life cycle. So bug is feeded. Bug is feeded, then through its feces contaminate or human, then in human it multiplies, and bug again feces, and cycle repeats. So here is it more detailed. Like trypanomastigote bug takes a blood meal, epimastigotes in midgut, multiply midgut, metacyclic trypanomastigotes in hindgut. Determined by bug takes a blood meal, passes metacyclic trypanomastigotes in feces, trypanomastigotes enter bite wound or mucosal membrane such as conjunctiva. Then in human, metacyclic trypanomastigotes penetrate various cells at bite wound site. Inside cells they transform into amastigotes. Amastigotes multiply by binary fission in cells or infected tissue. Intracellular amastigotes transform into trypanomastigotes, then burst out of the cell and enter the bloodstream. Then they or ingested by bug, or trypanomastigote can infect other cells and transform into intracellular amastigotes in U-affection sites. Clinical manifestation can result from this ineffective cycle. Transmission, mainly by contamination of skin abrasion by winged bug feces, but rarely may be contamination through infected blood transfusion, through infected mother milk, and through the placenta. Pathogenesis. First, acute form. Chagoma occurs at the site of a bite. Parasite reach regional lymph nodes to blood, then to organs and tissue. Fever, enlarged lymph nodes, skin rash, enlarged liver and spleen occurs. Romano sign may be unilateral conjunctivitis appears suddenly together with edema of upper and lower eyelids and cheek, like here. Then, or death due to meningoencephalitis and heart failure, or it passed to chronic form. Chronic form. Parasite produce antigen similar to patient self-antigen. The body produces autoantibodies that cause damage to heart muscle fibers, congestive heart failure, esophageal muscle fibers, megaesophagus and dysphagia, colon muscle fibers, megacolon and constipation, CNS or thyroid gland, exacerbation of infection in immunosuppressed patient. Diagnosis. Clinics plus finding parasite in blood film, biopsy from lymph node, liver or spleen, culturing, xenodiagnosis, serological test, cruising test, molecular techniques. Xenodiagnosis is highly effective demonstrative lower level of parasite in blood. Method: a laboratory bred winged bug is starved for 2 weeks, then fed on suspected patient's blood. 30 days later, it feces and gut examined for trypanosomes. So it's again finding in the blood, in biopsy, Romana Science, Chagoma. Clinical presentation. In chronic stage, these parasites resides in heart and midgut and sometime in hindgut, causing gigantism of the midgut, like here. Treatment. In early stage of sleeping sickness, again just repeat it, pentamidinofusp. In late stage, trypanosomide. For birth only lowest stage of the disease, eflorantine. For Chagas disease, nifurtimox inhibits intracellular development, drug of choice in acute and early chronic, or primaquine destroys trypanosoma in blood. Control. Treatment of patients, control of factors, and in sleeping sickness, pentamidine may be used in prophylactic drugs. In Chagas disease, elimination of reservoir host. That's all for the second part of the lecture. And now third, the last part of the lecture. It's about leishmaniasis. Leishmaniasis is a group of protozoan diseases of human and animals caused by different species of the genus Leishmania, characterized by affecting the internal organs, visceral leishmaniasis, or mucous membranes and skin leishmaniasis. It refers to diseases transmitted by mosquito of the genus Phlebotomus, leishmaniasis of the Old World, and Lutzomia, leishmaniasis of the New World. Leishmaniasis take an important place in tropical pathology. According to WHO, leishmaniasis is common in 88 countries of the world. In 32 countries, it is subject of obligatory registration. According to expert estimates the number of patients with leishmaniasis in the world, more than 12 million people. There are 2 million new cases annually. Approximately 350 million people live in endemic area. Leishmaniasis included in the special program of World Health Organization study and struggle with tropical disease. Leishmaniasis is one of the most actual problems due to its ability to suppress immune system, especially in persons with HIV or AIDS. Classification. According to ICD-10, B55 leishmaniasis, B55.0 visceral leishmaniasis, by type anthroponotic, Indian visceral leishmaniasis, zoonotic, Mediterranean, Central Asia visceral leishmaniasis, children's color other, in East Africa leishmaniasis, visceral leishmaniasis of New World. By duration, acute, subacute, chronic, latent, subclinical. By periods, initial, climax, terminal, cachexia. By severity, mild, moderate, and severe. Leiden syndrome, exanthema, fever, polylymphadenopathy, hepatosplenomy, hemorrhagic syndrome, cachexia. B55 dermal leishmaniasis, B55 mucocutaneous leishmaniasis. By type, leishmaniasis of the Old World, anthroponotic type, zoonotic subtype. Pindesh ulcer. Primary leishmanioma, secundary leishmanioma, diffuse infiltrative leishmanioma, tuberculoid lipoid leishmaniasis. Leishmaniasis of New World, American, Mexican, Amazon diffuse, Panamanian, Peru, Venezuelan. B55.9 leishmaniasis of unknown origin. Example of formulation of diagnosis. Indian visceral leishmaniasis, chronic form, moderate severity, manifest period with hepatobiliary syndrome. Geographic distribution of visceral leishmaniasis. So it's this zone is some in Africa, part of India, South America. Geographical distribution of cutaneous leishmaniasis more in Arabian country and more in South America, different part of Africa. Distribution of visceral leishmaniasis in Asia and Mediterranean due to Leishmanioma donovani and Leishmania donovani infantum. So in India it's both infantum and donovani. no reported case from these areas. It doesn't mean it doesn't exist, it means it's not registered. So we don't know for sure Leishmania donovani infantum in China, in Arabic countries, and Leishmania donovani partially in some of these countries. Distribution of visual leishmaniasis in Europe due to Leishmania donovani and Leishmania donovani infantum. So in Europe, it spread to mostly infantum and donovani in Italy. So from these countries, it's not, again, no recorded cases, but we have local leishmaniasis in Crimea several years ago. In Yalta, we have like almost subtropical climate. So while this lecture was made, it's no recorded case. I don't know, do we send this information to WHO organization, or did they count if it's only one case? But as one case was registered, there may be more cases, but just not registered. Distribution of visual leishmaniasis in Africa due to Leishmanium donovani and Leishmanium donovani infantum. Again, in most parts donovani infantum, in some parts mixed, and only in one country only Leishmania donovani. No recorded cases in these parts of Africa, but again, no recorded means just not registered. Maybe it present there, maybe really absent. Distribution of visual leishmaniasis in South and Central America due to Leishmania donovani chagassi. There is 90% of cases of visual leishmaniasis occur in the Southeast, Central Asia, Mediterranean, Sudan, and Brazil, and 90% of cases of dermal and mucocutaneous leishmaniasis in South America, North Africa, Syria, and Iran. Etiology. Type, protozoa. Subtype, Sarcomastigophora. Subclass, Mastigophora. Class, Flagella. Family, Trypanosomidae. Genus, Leishmania. Types, Leishmania donovani, Leishmania tropica, Leishmania mexicana, Leishmania brasiliensis. The cycle of Leishmania consists of phases. Amastigota develops in the human or animal body. Promastigota develops in organism of the mosquito or sandfly. It's such a trick. In your textbook and everybody it's written sandfly. In Russia, like there is no such word sandfly, it's only mosquito. So if you check Wikipedia, so English article will be sandfly, the same article in Russian will be mosquito. Yes, so in Russian lectures or something, it's often called mosquito, but it's the same as sandflies. So in human, promastigotes are phagocytized by macrophages. Promastigotes transform into amastigotes inside macrophages. Amastigotes multiply in cells including macrophages of various tissue. Then sandfly takes a blood meal, ingest macrophages infected with amastigotes, ingestion of parasite cells. Amastigotes transform into promastigote stage in midgut, divide in midgut and migrates to proboscis, and sandfly takes a blood meal and injects now promastigote stage into the skin. Then cycle repeats. Visceral leishmaniasis, natural focal transmissible protozoal infection caused by a special view of protozoa. Disease is characterized by affection of reticular endothelial system, clinically prolonged course, undilating fever, splenomegaly, progressive anemia, leukopenia, thrombocytopenia, cachexia. Visceral leishmaniasis is a typical parasitic reticular endotheliosis. There is distinguished Indian visceral leishmaniasis, black illness, dum-dum fever, Indian Kala-Azar leishmania disease, and other names of this disease, Mediterranean Asian visceral leishmaniasis or children leishmaniasis, East African visceral leishmaniasis, East African Kala-Azar, American visceral leishmaniasis of New World. Epidemiology. Source of infection, sick man. In Africa and other regions may be animals. Vectors, mosquito type of Phlebotomus argentypes. Vector becomes contagious in 6-8 days after blood suction when leishmania finishes its life cycle, and person infect after mosquito bite or sandfly bite. Besides the transmissible ways, there are possible non-additional ways: blood transfusion, sexual contact. Susceptibility of population is general, but young people 10 to 30 years in rural areas are more sensitive. Life cycle. So sandfly vector with promastigotes, wild canid reservoir or human host with amastigotes, and dog is reservoir too. So all this may be, so it may be to foxes, then to mosquitoes again, to humans, and again to mosquito, or to dogs. General pathogenesis. Gates of infection: skin. In a few days or week of bite of infected sandfly, small papilla or ulcer has appeared with multiplicating leishmania. Then the parasite spread hematogenically throughout the body and fix in the bone marrow, lymph nodes, liver, and spleen. Clusters of leishmania cause proliferation of connective tissue in paranephromatous organ, development of nephrotic and degenerative process, clinically enlargement of liver, spleen, lymph nodes, and disorder of their function, bone marrow failure, and GIT. Defeat of the blood-forming organs leads to severe leukopenia by reducing the number of granulocytes and progressive anemia. Reduced life of leukocyte and platelet. Clinically, necrotic tonsillitis, necrosis of the mucous membrane of the mouth and gums, NOMA. Products of metabolism and destroyed leishmania leads to the development of intoxication, clinically, fever, weakness, fatigue. Slow accumulation of specific anti-leishmania antibodies, immunoglobulin M earlier and immunoglobulin G later, and production of antibodies to the outer antigens. Clinically, it leads to activation of different autoimmune disease and immunopathological reaction. Kind of leishmania cause different clinical syndrome, has different geographical distribution, source of infection, and main vector of transmission. So vectors are more or less the same. Leishmania donovani cause visceral leishmaniasis, spread in India, Pakistan, Nepal, West Africa, China. Source of infection: rodents, dog, man. Leishmania infantum, visceral but mostly among the children, spread in Mediterranean, Central Asia, China. Reservoir: dogs, fox, jackal, dog. Chagas. Leishmania chagas identical to Leishmania infantum. Visceral, South and Central America is where it spread. Reservoir: dog, fox, opossum, man. Leishmania archibaldi cause visceral leishmaniasis, spread in Eastern and Central Africa, Sudan, Ethiopia. Reservoir: rodents, carnivores. Clinic: Indian visceral leishmaniasis or Kala-Azar. Incubation period from 3 weeks to year, on the average 6-8 months, rarely up to 2-3 years. Initial manifestations of the disease are often unnoticed. There are observed mild, moderate, severe forms of the disease and 3 periods of clinical course: initial, climax, acute manifestation, and terminal period. Initial period: general weakness, loss of appetite, adenomia, insignificant enlargement of the spleen. Climax period: fever is a cardinal symptom of the visceral leishmaniasis. Temperature curve is usually undulating, fluctuates to 39-40 with remissions. Duration of fever from several days to some months, of remission from a few days up to 1-2 months. Hepatosplenomegaly is prominent syndrome, permanent sign of Indian visceral leishmaniasis. Spleen is solid and takes the most part of the abdominal cavity. Splenomegaly develops rapidly, particularly on the 3rd-6 months of the disease, then it increases slowly. Enlargement of the liver is usually less significant. Palpation of the spleen and liver is dense but painless. Pain can appear due to development of perisplenitis and perihepatitis. Lymph nodes are not enlarged. Skin gradually becomes dark or black color, synonym black disease, due to decreased function of adrenal glands. Hair loss, the shine, becomes dry and brittle. Loss of body mass, anemia, granulocytopenia, and agrogranulocytosis. Necrosis of the tonsils, mucous membrane of the mouth and gums, NOMA. Cardiovascular syndrome, heart shift to the right due to hepatosplenomegaly, cardiac tones become dull, blood pressure is decreased, tachycardia appears in the feverish period and continues in remission. With the development of anemia and intoxication, signs of heart failure is increasing. Hemorrhagic syndrome, ascites and peripheral edema is developed due to portal hypertension and hypoalbuminemia. Secondary infection can be activated due to agranulocytosis, possible diarrhea in women, oligoamenorrhea. Terminal period, cachexia, sharp hypotonia of muscles, skin is thin, through the abdominal wall can see the borders of enlarged spleen and liver. Prognosis of mild form of visceral leishmaniasis is favorable, but in severe and complicated form serious. In HIV patient, visceral leishmaniasis has malignant character and is accompanied by resistance to specific therapy. In CBC, anemia up to 50 gram per liter, leukopenia up to 2.5 x10 in 9 degree and less, neutropenia and relative lymphocytosis, uneosinophilia constant site, coagulation of blood and erythrocyte resistance in decrease, ESR sharply increased till 90 millimeters per hour. 5-10% of patients have skin leishmanoid, which develops in 1 to 2 years after effective treatment and consists of macula, nodules, papilloma. Leishmania contained in leishmanoid can survive many years. Thus, a patient with skin leishmanoid, reservoir of the agent and the source of invasion of mosquitoes for many years. So it's a symptom according to days. Anorexia, almost a month. Adenomia, a bit less, but still almost a month. Hemorrhagic syndrome starts from 4th day and lasts again almost a month. Anemia lasts a month. This puncture of bone marrow was present and treatment was given on the 23rd day. So on the basis of treatment, in a week, symptom stops. Several hemotransfusion was made. Body mass decreased, then increased. Temperature, like high, normal, and then normalized in a week. Hepatomegaly was from the start and become less expressed, and splenomegaly lasts longer. It was a specific case, and its symptoms. Mediterranean Kala-Azar. Most cases of co-infection are registered among men aged 20 years. 25-70% of HIV infected and 1.5 to 9% of AIDS patients suffers from visceral leishmaniasis. That's why visceral leishmaniasis in HIV infected is the most frequent cause of opportunistic infection. Hematogenic syringe, where is easier realized due to high concentration of leishmania in the blood of HIV patient. The most significant features of Mediterranean AIDS and visceral leishmaniasis are incubation period within 1 month to year and average 3-5 months. Skin leishmanoid is absent. Involvement in pathological process peripheral and visceral lymph nodes with the development of mesedenitis. Frequent development of hemorrhages and bacterial pneumonia. Hyperpigmentation of skin is absent. Skin is pale, gray colored. Primary effects as papules, sometimes covered with scale before the manifestation of the disease. The disease may occur in acute, subacute, and chronic forms. Patient with leishmaniasis. The East African visceral leishmaniasis. Leishmania archibaldi, East African Kala-Azar. Sources of infection: wild mammals, of the attachment of rodents and carnivores. Vectors: mosquitoes of the genus Phlebotomos, Phlebotomos orientalis, Phlebotomos martini. Suspicible person of all age young people living in rural areas. Pathogenesis is similar to other visceral leishmaniasis. Peculiarities of clinic. Symptoms are similar to Mediterranean Asian visceral leishmaniasis. More frequent development of primary effect in the form of papules and ulcers. Skin leishmanoid appears after clinical recovery. Tendency to relapse of the disease. Visceral leishmaniasis of New World. American, South American visceral leishmaniasis, Leishmania chagassi, identical to Leishmania infantum. Epidemiology is similar to the visceral leishmaniasis of Old World, caused by Leishmania infantum. The causative agent of American visceral leishmaniasis has typicity to the canids, is registered in Central, South America, Mexico. Vector: mosquito Lutzomia longipalpis, which actively attacking dogs than people. Pathogenesis, clinical symptoms, complication, diagnostic, treatment are similar to other forms of miscellaneous leishmaniasis. Prognosis in symptomatic cases is serious. There is a large number of asymptomatic disease with presence of leishmania in the skin. Prevention is the same, includes active detection and treatment of patient, the destruction of sick dogs, fight against mosquitoes. Differential diagnosis of Indian, Mediterranean, Asian, and East African. In Indian, pathogen Leishmania donovani, age of patient 10 to 30 years, affects lymph nodes, liver, spleen, bone marrow, GIT, epinephral gland. Primary effect is absent. Incubation period 6 to 8 months in average. Peculiarities of course of disease: initial, peak terminal, fever from days to months, spleen from 3 to 6 months, hypersplenism, liver less than spleen. Mediterranean Asian pathogen Leishmania donovani infarctum. Mosquito is contagious in 6-8 days. Age of patient 1 to 5 years. Affect most lymph nodes, peripheral, peribronchial, mesenteric. Primary effect in 1.5 year. Incubation period from 22 days to 3-5 months. Course acute in small kids, rare subacute 5-6 months, lingering in adults. Outcome is favorable. East African pathogen Leishmania archibaldi, age of the patient young, affects the same as in Mediterranean Asian. Primary effect more often, possible ulcers, incubation period up to 3-5 months, particularities of clinical course, tendency to relapse. They all have the same vectors, mosquito, phlebotomus, or sandflies, the same but different name. In 6-8 days, mosquito becomes contagious. Fever from days to months to oldest form. Spleen in East African moderate. Liver less than spleen in all cases. Again, other scenes. East African skin with earthy shades. Necrosis not always happen. Hemorrhagic syndrome, ascites, diarrhea is present. Pneumonia may be present, may be absent. Skin leishmanoid is absent. Mediterranean Asian skin is porcelain. Maybe noma, necrosis of tonsils, mucous membrane glands, hemorrhagic syndrome, ascites, diarrhea, and pneumonia present. Skin leishmanoid is absent. Indian skin is black. Necrosis and noma may be happening, like tonsils and mucous membranes. Hemorrhagic syndrome present, skin GIT nose, ascites present, diarrhea may be present, may be absent. Pneumonia may be present, may be absent. All of them have remission from several days to 1 to 2 months. Blood, in all cases, anemia, leukopenia, thrombocytopenia, and in Indian, skin leishmanoids in 1 to 2 years after treatment may appear. Diagnostic, the same for all. CBC, ELISA, RLA with antigen leishmania, biological assay, infection of hamster, punctate culture, test for obtaining a positive culture on a specific medium, NNN, prevention, identification of patient, CBC, ELISA, RLA, ensanation, anti-money drugs, amphotericin B, control of factors, sandflies. Diagnostics. Parasitological method, revealing of leishmania in the blood, smear and sick drop, biopsy, puncture of bone marrow where leishmania easily detectable. Sometimes trepanna biopsy of iliac bone or biopsy on the lymph nodes, rarely of spleen and liver can give positive result. Punctate culture test for obtaining a positive culture on a specific medium. Serological investigation, CBC, ELISA, and ELISA and relay with antigen leishmania. Biological assay, infection of hamsters. PCR and express method, immunochematographic detection of recombinant clone antigen Ki39. In Mediterranean Asian visceral leishmaniasis, serological and intracutaneous tests are non-informative. We should remember that in half of HIV patients with visceral leishmaniasis, serological test will be negative. In 75% of patients, leishmania will be revealed in the blood, and in 88% in the skin and perineural fluid. Treatment. For the treatment of visceral leishmaniasis, use drugs of 5-valent antimony, sulosurmine, pentostem, sodium stibogluconates, glucantin. Synonyms: Megalomenis tibias. Contradiction: pneumonic tuberculosis, severe violation of the liver and kidney function. Side reaction: fever, cough, vomiting, polyneuritis, myocarditis. Introduction: intramuscular deeply. Daily dosage: 0.06 gram per kilogram of body mass. On the first day, 1/4 of necessary dose. On the second, a half of necessary dose. And the third day, 3/4 of necessary dose. On the fourth day, full dose. The course of treatment is 10 to 15 injection. Repeat a course through the 6 weeks. At non-effective therapy with antimony drugs, appoint amphotericin B and paramycin. Aminazidine can be administered simultaneously with drugs 5-valent antimony in 2 syringes in different places. For the treatment of visceral leishmaniasis can also be applied pentamidine 4 mg per kg per day during 11 weeks, amphotericin B full dosage of a course 20-30 mg per kg during 10-20 days. In addition to specific treatment is necessary pathogenetic therapy and prevention of secondary bacterial infection. It is necessary to widely use restorative therapy, drugs, iron and phosphorus per os, vitamin B12, diet rich in animal proteins and vitamins. Treatment of post-Kala-Azar skin leishmanoids is the same as visceral leishmaniasis, but the course of treatment continued to 2 to 4 months. The first oral medication for the treatment of visceral leishmaniasis, miltefosine, has been registered in India. It is suggested that miltefosine will replace drugs of 5-valent antimony and amphotericin as drugs of first line. The fullness of recovery is estimated by next criteria: significant improvement of the patient's general condition, normalization of body temperature and absence of increase during next 1.5 months, decrease of liver and spleen sizes, morphological normalization of blood picture, noticeable increase of hemoglobin and albumin, decrease of gamma globulin level, absence of leishmania amastigota in the repeated investigation of sternum punctate, identified by Romanovsky-Gimze. Be healthy and happy. That's the end of the lecture. Thank you for attention.